board. So today's February 24th, 2021. It's the first day of our new unit. And today's title is DNA structure and function. We'll touch more on the DNA molecule, which we have talked about before. I am going to try to use this whiteboard for the first time. Why not use it? The objective today is to analyze the structure and function of DNA and better understand how it is synthesized. And your essential question, how is the structure of DNA related to its function? Okay, I'm going to use my folks who I can actually see as indicators that it's time to move on. We've got until 12:30. We've got a long time left. Okay. I'm going to jump to the next slide here. So midterm, like I said, I expect it will be Tuesday, March 9th. They have not yet released the final schedule. But whenever it happens, it's going to focus on these 15 standards that we've covered. The purple standards on the right side are the standards that we're covering in this unit. DNA structure and function, protein synthesis. We'll talk more about, what'd you say? No, you don't need to write this down. We'll talk more about mutations, and then we'll talk about the process of cell specialization. That's one of my favorite standards. So it'll be a fun unit. I think now we can actually start to have some more sophisticated conversations, and we'll talk about some of the technologies that are coming up and making this process a lot more manipulatable, more easily manipulated is what I'm trying to say. Okay, so I'm big on mind maps. You guys have heard me say that before, but now that I can actually see you and check and see if you're actually writing things down, we're in a good spot to actually do one of these mind maps. So a mind map for me is an opportunity to connect ideas and topics that might seem like they're not easily connected. So what I want to do now, and if you actually pin like me talking to your screen, you click on pin to screen, then it'll make it bigger. And hopefully you all will be able to see what I write on the board. We've already talked about DNA. We've already learned about it a little bit. So now I want you all to tell me what you remember about this molecule. Anything. Is it backwards on your screen? No, okay. What do we remember about DNA?
It's made of nucleotides.
Good.
I think it was it's packed neatly, like it's like in order.
I think that was Lisa. Was that Lisa? Thank you, Lisa. I like that. So I wrote the order, aka the sequence, matters. And then we've got another comment from Abigail. They have the instructions to make proteins. Good. DNA contains the instructions to make proteins. You're close, Janelle. It's not ATUG. There's another letter that's not U. ATCG. You see that? Let's go down a little bit. Yeah, so the nucleotides have 4 different nitrogen bases. A, T, C, G. Notice how I paired them up. I did write that like that specifically, intentionally. I'm sorry I skipped over yours this way. Yeah, DNA is found in the nucleus. Is it found in the nucleus of every cell, or is there a specific cell type?
And eukaryotic cells is found in the nucleus.
Wonderful, thank you. I'm pretty sure that was Janelle. Found in the nucleus of eukaryotic cells. What about prokaryotic cells? Where might we find DNA in a prokaryotic cell? Does anyone remember? Good, Janelle, yeah, and Dijon, excellent. It's found in the cytoplasm. Which of the 4 macromolecules is DNA?
Nucleic acid.
Yes, thank you. So I've got 2 things to write down. And then, oh yeah. Oops. Okay, I'm going to circle some letters here just to give us mental reminders that these are related terms. So we see these letters, these 5 letters, N-U-C-L-E, coming up in several places. So when we see NUCLE, hopefully we're starting to make this connection that we're talking about something related to DNA. Okay, I think this puts us in a good place to keep going. This will still be available to you all. I'm not going to erase it right now. But you can unpin me if you want to so that the other screen will come up. Gotta unpin myself. How do I do that? There it is. All right, so DNA structure and function. DNA stands for deoxyribonucleic acid. And it is a type of nucleic acid, the monomers of which are nucleotides. So we basically just wrote this down. If you already have it, perhaps what you should just write down is what DNA stands for. Deoxyribonucleic acid. Funny enough, we really didn't write down the structure of DNA, nor did we explicitly say what its function is. Does anybody remember what the shape of DNA is called? What kind of helix? Okay, there's another word that goes in front of helix. There's an adjective. Not one, but not single, but what'd you say, Bina? I thought I heard you say something. Double, thank you, Dijon. It's a double helix. Oh, I didn't mean for that to happen. So there's your double helix structure. It's basically a spiral, as Lance said, or we can think of it as being a ladder that just gets twisted. I'm going to refer to that analogy as we go forward. And the function of DNA, of course, is to store genetic information. This is how cells can pass on specific traits. Just because prokaryotic cells don't have nuclei does not mean that they do not have DNA. As Janelle told us, prokaryotic cells do have DNA. It's just found in the cytoplasm instead of in the nucleus. So all living things have DNA. At least all currently, or all life that we're currently aware of has DNA. Okay. I can see some people are still writing, but what? There's no way. Ready? Okay, so you don't need to actually reproduce this drawing. But I just wanted to highlight the different parts. We are going to draw something similar. And because you all are my honors class, we're going to take it up a notch as well. But we can see that this structure of DNA is really shaped like a ladder. But DNA has an important feature, and that feature is that it is what we call antiparallel. I'll talk more about that. But we can see that there is a nitrogen base, and those nitrogen bases can be adenine, which we use the letter A for, thymine, which we use T for, guanine, which we use the letter G for, and or cytosine, which we use the letter C for. Then there's a 5-carbon sugar. We'll talk about why it's called a 5-carbon sugar, but it's called deoxyribose. Deoxy because it's actually missing an oxygen. And of course, sugars end in those letters. And then lastly, there's a phosphate group as well. So now I want to actually draw it. So I'm going to erase what's on the board. I'll put up the first image that I want you guys to draw. It's just a pentagon. And inside of the pentagon, just write 5-carbon sugar, or you could just write deoxyribose. Does anybody still need this? That's fine. I always imagined that you guys would be my more talkative class. So like I said, you guys are my honors class, so we're going to go a little bit deeper into the chemistry of this. It's a 5-carbon sugar, which means, of course, that it is made up of 5 carbons. So we can actually number the carbons. There's your first one. It's on the right corner. Then your second one is in the bottom right corner. Okay. Your 5th carbon is not actually within this pentagon. It's not in the ring. So that can be a little confusing, but it is still connected to the ring. It's just up there. It's kind of like a flagpole. The reason we number these carbons, yes, they are the same atom. They're made of the same element. And for those of you who are here, if you look behind you, there's a periodic table of the elements. You can see all those different elements. They each have different chemical properties. They boil at different temperatures. They, have different, they make connections with different elements. Carbon is number 6 on that list. It's probably the most important element. In fact, it's the element that really gives us life. So even though these 5 carbons that are a part of this ring are the same thing, they have the same chemical properties, they have the same number of protons and neutrons and electrons, we still need to number them because they give us direction. So this is number 1. That tells you which way the sugar is organized. The fact that this one is number 3 and this one is number 5 will become very, very important shortly. The tip or the top of the ring is an oxygen atom. Oxygen is number 8 on the periodic table. So what you have in reality, even though I wrote them on the outside of the ring, in reality, these are all connected. They're bonded to one another. that probably? Okay, that didn't come out how it should have. I apologize. But I'll draw it on the board so it looks a little bit, it makes more sense. There's this phosphate group that is connected to the fifth carbon. Again, the fact that it is the fifth carbon is important because the fifth carbon will always be the one that is connected to the phosphate group. So that looks a little funky, I know, if you're looking at the screen, but it should be PO4, and the 4 should be a subscript. So it should be kind of written on the lower level. And then a superscript outside of the parentheses should be -2. So that's just a chemical compound called phosphate. What it's saying is that P is the element phosphorus. It's number 15 on the periodic table. And that P is surrounded by 4 oxygen atoms. That's why it's O4. And because it has so many negatively charged oxygen atoms surrounding it, it ends up with a chemical charge of -2. This will make more sense when you all take chemistry. But I just wanted to give you an idea of what it looks like now. The last thing is, of course, the nitrogen base. And the nitrogen base could be A, T, C, or G. Notice that the nitrogen bases are connected to the first carbon. So again, it's important that we can number these carbon atoms. Okay. One last thing, kind of. So again, the numbering is important because it gives us direction. So these nucleotides are, and I've always imagined them like an arrow. The oxygen is pointing in the direction that the nucleotide is kind of moving. It's moving from the 3' carbon, from the third carbon, towards the 5' carbon, the fifth carbon. And what you end up with is this allows you to see how the nucleotides get bonded to one another. So I'll show you what I mean by that. So you end up with longer and longer chains of these nucleotides, and they're connected to one another at this 3' carbon. They're connected to one another at the 3' carbon. So we could even draw another one up here if we wanted to. That's the other way. Okay, any questions about this? Any questions from you folks at home? Okay. So what we end up with is that the sugar and the phosphate serve as the backbone. They're like the sides of the ladder. Whereas the nitrogen bases are the rungs of the ladder. Oops. Okay. Yes, you can. So I wonder, was I moving too fast when everybody was at home? Because now I'm sitting here, I'm like, oh, okay, it takes a little bit more time than I thought. All right. Was that enough time? Okay. All right. So what do we notice here? Let's be sleuths. Let's be detectives. Look at the colors. What are you noticing about the colors of those nitrogen bases in the middle? They're together, and they're together in what way? What's the pattern? Somebody's trying to say
purple, green, then blue and pink, and then green and purple.
good. So good, and thank you, Abigail, for that contribution too. The purple and the green are always together. Now the colors really don't matter here. The colors are arbitrary. What we need to focus on is the actual names of the bases. So adenine and thymine are always together. A and T always get paired up. And then we can also see that the pink and the blue, again, the colors don't matter. It's the cytosine and the guanine, the C and the G, that are complementary to one another. They always pair up, C and G. So that's going to be on the next slide. I'm just going to keep going on to the next slide. Pin this really quick. Hopefully that works. Lance, we just discovered that the A and the T always pair up, and the C and the G always pair up. Did you know that already? How is that visible? Okay, so excuse me, my pentagons are not perfect here on the board. I'm going to move in a little bit closer. They're not perfect, but we can at least notice, we should be able to notice, that they're pointing in opposite directions. So over here, they're running this way, down, they're pointing that way. Over here, they're running up. And if you remember from our original drawing, I told you that the oxygen is always going to be pointing up. The tip of the pentagon is always going to be pointing up. And I also told you that it points in the direction of the fifth carbon, 5 prime. So 5 prime is always going to be the tip of the arrow. So 5 prime is always going to be the tip of the arrow, which makes sense if you think about it. In my mind, 5 is the highest number of the carbons, and so it points. It's the top carbon. It points at the top of the arrow. No, it's not. I can't. Yeah. Hopefully it's not converted. Okay. So obviously these are running in opposite directions. This one is 3 prime to 5 prime. That one is 5 prime to 3 prime. So this is a concept that we call anti-parallelism. Yes, they're parallel to one another, meaning they run in the same direction. I mean, they're lined up with one another, but they're anti-parallel because they run in opposite directions. It's just like, most city streets are anti-parallel. You've got people traveling on the right side in one direction, and then people on the other side of the street are going in the opposite direction. Same way that DNA runs. Okay, now let's see. All right, so as you can see on the board now, I've got 4 nitrogen bases. I've got A, C, C, D. Now notice over here, we've already talked about how they are complementary to one another. So which nitrogen base should go right there? That should be a T. And then what about there? And then what's next? C. And then C. And then those nitrogen bases are bonded together as well by hydrogen bonds. Bless you. All right, I'm gonna wheel over here. Scoot in a little bit. So let's have that. I've got two strands of DNA, two molecules of DNA. One of them is TAA, CGG, CATT. The other is TAA, GCG, CATT. What's going to be the complementary strand on the left side? Which should come first? A, T, okay, what about on the right side? Let's have somebody else. You clearly got it. And you too, JT. Both of you clearly got it. So somebody else help me with this one on the right side. A, T, T, C, G. Excellent. All right. So very similar strands of DNA, very similar molecules of DNA. There's only a slight difference here. Where is it? Oh, and I messed up here. Sorry. It should be a C. Where is it? Where's the difference? Yeah, here it is. So even though we only have a difference of two nucleotides, there are only these two molecules are different in only two places. That would create an entirely different protein. So small changes, this is what I want you guys to understand, and we'll explore this more when we talk about mutations. Really small changes in DNA have big impacts. And that's because the sequence matters. Even if I have three nucleotides, ATT is different than TAT. ACG is different than AGC. Okay, so the order matters. The way that they are arranged next to each other plays a big role in what the actual molecule becomes. We'll talk more. 7 more minutes. I think only a couple slides left. What does that say? Okay, we talked about this. So if you guys wrote this down when we did the word map, you don't need to write it down again. But DNA, of course, is found in the nucleus of eukaryotes. Janelle told us that earlier. And it is found in the cytoplasm, of prokaryotes. We're going to talk more about chromosomes as well. Plasmids don't really come up all that often. So we will talk about plasmids once more in the course, but not very important. Is anybody writing here? I'll wait a little bit. Okay. So it's the order, again, it's the sequence of nitrogen bases that matters. We're going to talk a lot more about that probably on Monday. I think we won't get to it until Monday, but this is because of a process called translation. So it's the order that really, really matters. Keep in mind, guys, that many of the things that make life possible are common throughout different forms of life. What this means is that the genes that make us humans are very, very similar to the genes that make chimpanzees chimpanzees. We both have central nervous systems, so we need the genes that code for neurons and glial cells and specific parts of our brain that can function involuntarily. We need those same genes. We also need circulatory systems, so we need genes that code for red blood cells and white blood cells and lymphatic systems. We need digestive tracts, So all these different things are coded for by the same genes, even though we end up being very, very different species. So we're still 98% genetically similar to chimps. Even a mouse, we're 92% genetically similar to mice. Even something that is totally unlike a human being, like plants, we share many genetic we share 1/5 of our genome with plants. So life itself obviously has come to take on a great amount of diversity. We'll talk about that later on in the course, but we have these similarities within our genomes that we see present in all forms of life. So it's pretty cool. Now, between any two human beings, we know that there are very, very few genetic differences. So we're 99.9% genetically identical to one another. So that 0.1% of difference is what accounts for all of the variation we see in skin color, hair texture, height, allergies, personality traits. It's only covered by 0.1% of our genes. Of our DNA. Now, there is another fun fact that says there that genes make up only 3% of your DNA. That's true. We're going to talk a lot about that. So if we picture it like like one of these textbooks in the back of the class, really, really thick book. If DNA is a thick book that has every single letter, AT, AT, CG, AT, GC, over and over again, there would be 3 billion letters, just A's and T's and C's and G's. But of that, there are only about 30,000 genes within that. So a gene is just a small section of DNA that can get carved out and that actually codes for a protein. Genes code for proteins. We'll talk more about that. Okay, we've got 2 minutes. So if you guys want to give it a try for the exit ticket, I think that'd be a good idea. There is no asynchronous assignment today. So all you need to do is knock out that exit ticket and then you'll be done. Thanks for paying attention today. I like the whiteboard. How do you guys feel about it? Okay, cool. Okay, so sorry guys, I didn't give you enough time when I was thinking, you can always pause it, right? Or just leave it pulled up on your Chromebook. We done? Cool. For those of you at home, thanks for being here. I will talk to you all tomorrow. I hope you have a good day.